Hi everybody, welcome to our lecture on membrane structure. We're going to split this one up. It's supposed to be membrane structure and function. We'll just start with the structure part because we had to catch up from last week. So here we go. Here are your learning goals. There's quite a bit, so please make sure you take a look at it and make sure you guys are comfortable with those learning goals and go a little bit deeper, right? So for something like differentiate the three types. Don't just list them all out. Like, how are they different, right? What are some things that I can compare them to? That kind of stuff, right? And then I did want to point out this one, the techniques that were used to study membrane proteins. We're not going to have some time to talk about it in class, but it is in the textbook, and I will add some supplemental videos for you guys to watch in regards to that, so it'll still be fair game. So, as always, we're going to start with a little bit of brain digging. So it's going to ask you to draw or write about what you remember from your previous studies about what makes up a membrane. So same thing, pause, time yourself for 3 minutes, try to see how much of it you can draw or words you can add, right? And then we'll go from there. Okay, so hopefully you came up with something similar to this, maybe not as detailed, but similar, right? So we have our membrane, and it's not just phospholipids, right? This is called a phospholipid bilayer because you have one layer here, right? There is the word semi-permeable. You might have put that on there, meaning that it can only allow certain things to go in and out. For our cells in particular, for animal cells, this is the only barrier we have against everything else, right? Plant cells, they have their cell wall, but us, this is the only barrier. So it can only allow certain things to go in and out. I would challenge you to think about the things, right? What are things that cannot come in, right? So think about that. See if you can figure out what that is, right? Maybe you came up with the word fluid mosaic. Fluid mosaic is just a type of art, right? So if you know what a mosaic is, it's the one where you have tiny pieces of like tile or rock or something. You put them together and you get this wonderful art piece, right? And the same idea here with the membrane. You have all of these tiny, tiny little pieces, put them together, and you get this magnificent, beautiful structure, right? We have these two words, hydrophobic and hydrophilic. I know just from last week we talked about those. You guys are aware of those. Remember, we do also have proteins in our membrane, so we'll talk about that today actually in a little bit more great detail. Same thing with glycolipids, right? So here is the structure of a phospholipid bilayer. The phospholipid ones are gray and the cholesterol is green, right? And I'm sure you guys remember again from last week we talked about this, but the head of our phospholipid head is hydrophilic, right? And then the tails are hydrophobic, right? So the middle part of our membrane does not want to interact with water. And the cool thing about these is actually if I have a handful of phospholipids and I kind of just toss them in water, they will immediately create their own ball of membrane almost, or a ball of lipids that looks like this. That's called a micelle, right? They do that automatically because just with the chemistry of their tails and their interaction with water, they don't like water, right? They don't like water. Their tails don't. So they orient themselves so that in a micelle, right, the heads are interacting with the liquids around and the tails are safe inside. And there was that word that we mentioned also, amphipathic. It just means that it has both hydrophilic and hydrophobic portions on the molecule itself. Cholesterol we'll talk about in just a little bit, but it is that green part that you see there in the membrane. Okay, so in 151, we've only really talked about phospholipids and cholesterol, but there's actually 3 types of membrane lipids, right? So we're going to go through each one of them, one by one. So first one is phosphoglycerides. So like I said, in our basic cell biology class, we've only talked about phospholipids, but actually on top of the phospholipids that we know, that's the one with the heads like this, right? On top of them, they have a glycerol backbone. So if you want to think of it as kind of like a hat almost, they have a little tiny hat, right? They have something on top of their head that characterizes them a little bit more. So then that's how we get the word phosphoglyceride, right? Glyceride meaning sugar, right? There's some kind of sugar on there. And there's 4 different types, right? I'm going to butcher these names. I don't know if you guys noticed, but I already stutter enough as it is. So we're going to break it down one syllable at a time. So there's here we go. Phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, and phosphatidylinositol. This is also partly why I cannot be a doctor. There's way too many big words out there. But they are abbreviated there for you. I am going to need you to know what they are written out, right? So it's the same prefix as phosphatidyl, right? And then whatever is attached to them. So if you take a look at the bottom structure here, and we'll just ignore that one really quick. It's the same thing. Here's your phosphate head, your 2 tails, and then the hat that goes with it. And you can draw different hats if you want. I don't really care, right? Tails, and then, I don't know, this one has a flower hat. Who knows, right? But that's what the structure looks like. There's something on top of the hydrophilic heads that characterizes them a little bit more. Depending on the phospholipid or the phosphoglyceride, right? They can be fully saturated, monosaturated, or polyunsaturated. When you guys were in 151, you might have just talked about saturated and unsaturated, right? But just like the word is telling you, monounsaturated means there's only one. Polyunsaturated means that there's more than one. So 1 plus. Right? Fully saturated, we'll talk about that in just a little bit. Let me just backtrack here a little bit because I forgot to mention it. So PS and PI actually has an overall negative charge. PC and PE has a neutral charge. Right? And you might be asking, what the heck is that for? Right? What the heck is that for? So if you have the head group, so the phosphate group, right, and you stick a hydrophilic group on top of it, right, the more hydrophilic that group is on top, the hat, right, the highly soluble the water domain becomes. So when it comes to PS and PI, right, the reason why it's negatively charged is because it plays a specific role in cell signaling, apoptosis, and it's actually found commonly in brain tissues, right? So the charge there is important, whereas PC and PE, they don't have that special negative charge, so they are probably more of a structural phospholipid than something that has an extra role to it, right? So those are phosphoglycerides. Next up, that is probably new to you guys, is sphingolipids, right? This is also a type of membrane lipid. It's just we don't use it and talk about it very often, right? So what is special about this is that it is a sphingosine, right? A sphingosine right here, right? It is just an amino alcohol with a long hydrocarbon tail. Now, if you've not taken organic chemistry yet, don't worry about it. It's not a big deal. But you can see there's a structure right here. So if I give you a structure like this, obviously it doesn't have these names on it, right? If I give you a structure like this and I ask you to identify what it is, your clue is it doesn't have a phosphate head, right? So I said that it had an amine alcohol. So here's the amine right here, alcohol right here, right? And then it's attached to the fatty acid. So that's a sphingolipid right there. So just like the phospholipids, they are also amphipathic, which means they also do have a hydrophobic and hydrophilic qualities. The main difference between this one and the phosphoglycerides or phospholipids, whatever you want to call them, is that they're fatty acyl tails. So these guys, the tails, right, are longer and more highly saturated. So in case you didn't catch this or you didn't make this relationship, right, saturated means that they're packed together, right? So if I have a membrane, I guess I should have explained this in the previous slide. That's okay. So if I have a membrane like this and all the tails are straight, that means that they are saturated. Now, there are some phospholipids that have that double bond that makes that kink. It does a little kick like that, right? So this one would be saturated, this one unsaturated, right? So for sphingolipids, their tails are longer and they tend to be straighter, which means that they're saturated, right? They're more packed in together. The other thing I wanted to point out is a glycolipid, right? So glyco, hopefully you remember, is a sugar. So if I have a sugar attached to a lipid, that's a glycolipid, right? If I had a sphingosine that is attached to a fatty acid, then it is a sphingolipid, right? Sorry, my words are all getting mixed up already. This is, again, why I don't teach too much medical terminology, because I stutter too much. But they are prominent, glycolipids are prominent in the nervous system and plays a role in certain infectious diseases, right? So you do have glycolipids sticking out of your membrane sometimes, but it is more common in the nerve cells, not really in other body cells, right? The last type of molecule lipid that we want to take a look at is cholesterol. If you'll notice, there's not a lot on there because we're going to talk more about it a little bit later. But essentially what it is it affects the fluidity of the membrane. So if I have, and I'm just going to draw one layer of the phospholipid here really quick so I can show you guys, right? Because it also affects the saturation, which I'm sure I'm probably getting ahead of myself again, but that's okay. We'll just talk about it again once we get to that portion. So if you think about each of these phospholipids as a kid in line for lunch, right? If you guys remember in high school or in middle school, you get in line for lunch, right? If they are just the students, right? Just the students, and let's forget social distancing for a little bit. If they're just the students, right, then you can pack a line pretty tightly, right? For my Disney people, you guys know how it goes. If there is a line, they'll try to like shove you in there as much as possible, right? But if for some reason, you guys might have experienced this before, if for some reason the person has its leg out, right, like, or they're like standing with their legs wide or something, then the line has a little bit more space to it, right? So that means you are not as saturated. And then sometimes, and this is very, very common around here in school or at Disneyland, if somebody has a backpack, so let's pretend this is a backpack, right? The backpack is synonymous to cholesterol. It's a very small molecule, and it embeds itself in between the legs of the phospholipid, right? So if you have cholesterol in your membrane, then you have space between you and the other person, right? So the more people you have a backpack attached to it, right? You have cholesterol attached to your phospholipid or right next to it. They don't have to be attached, right? Then the more fluid your membrane becomes, right? Because there's space. Fluid just means that there's space for movement. Right, so again, we'll talk about cholesterol in just a little bit, but this is the structure of that. Right, so then if we take a look at the phospholipid top and bottom, they're actually not mirror images. I know sometimes when we draw it in class or we just model it out, we think that it's exactly the same, right? But they are not. They are different. So here is a figure from your textbook, right, that shows you the composition of each of those. So these are all your phosphoglycerides. This is your sphingolipid and your cholesterol, right? You can see that they kind of balance each other out, right? So if there's a lot of sphingomyelin or sphingolipids on the top, not so much on the bottom, right? And then same thing with like PE, right? Lots of it towards the cytosolic side, but not so much in the endoplasmic side, right? So main idea, they are not exactly the same, right? Not exactly the same. Okay. So remember, we're taking a look at the different compositions that make up the membrane. So one of the things that you guys have on there, let me go back to the lipid here, is this one, glycoprotein, right? Glycoprotein. So a glycoprotein, like it sounds, it's a protein. Glyco means sugar, so you have a sugar attached to your protein, right? So that happens through the process of glycosylation. So you have a protein, right? So again, this one is sugar, right? Sugar. And what that sugar is called an oligosaccharide, right? So you have a protein, here's my protein, and then you have like a chain, this is going to be ugly, but a chain of carbohydrates on top like that. That is your oligosaccharide. And so the purpose of these glycoproteins is this bullet point right here. So it's important for cell-to-cell interaction, cell membrane identification, and sorting of membrane proteins. So what I usually use as an analogy is like your ID card, right? Now, in college, we don't really carry our ID cards anymore as we do in high school, but it's like your license, right? It's how your cells know that the cells that belong in your body are the ones that actually belong there. That's how they know that it's a virus, or it's a, when you're doing transfusion, that's how you know that it's not blood that belongs to you. That's how they know to attack it, because your cells don't have eyes, right? So what they do, let's take the white blood cell as an example. The white blood cell, what it does is it kind of grazes against a cell, takes a look at the glycoproteins or the sugars that are sticking out of the cell. If they determine that, hey, this is a protein or a cell that's supposed to be here, they leave it alone, right? But if they brush up against that sugar and they realize that it is a foreign entity, then that's how they mount the immune response, right? So an example of glycoproteins are the ones that you see on the edges of your red blood cells, right? This is how we determine what blood type you are, because of the glycoproteins, the sugars that stick out of your membrane, right? And this is, again, how your body knows that you're being given the wrong type of blood, is because the red blood cells will find that, right? Will find this blood, whatever it is, and if it doesn't belong in your body, then they start attacking it. So that's when like things like organ rejection happens, right? Because your body knows this liver or this kidney does not belong to you, so I'm going to start attacking it. Obviously, that's not what we want when we do organ transplants, but that's what it's for, right? That is the purpose of the sugar on top of your membrane. So here's another picture of the fluid mosaic. One of your goals is to be able to explain why it's called fluid mosaic. But again, the main idea is that it's just made up of a lot of different things, right? And if you look up what a mosaic is, I probably should have added a picture on here, but if you look up what a mosaic is, it looks exactly like that, right? So I can give you a blank diagram like this, and I could ask you to label it. That is very, very fair for me to do. So make sure you know how to do that, and make sure you're able to determine what the functions of those things are. And if you ever see one that you're like, we have never talked about this, then obviously I'm not going to ask you about that, right? So it's just all the ones that we've talked about or you're seen in this picture. Okay, so we've talked about the membrane, we've talked about the membrane, we've talked about these green things that are sticking out, that's your oligosaccharides. Now we're going to talk about the proteins that are embedded, right? So if we take a look at this photo, it's going to be those red kind of lumpy globular things, right? There's 3 types of membrane proteins: integral, peripheral, and lipid anchored. You should already be familiar with integral and peripheral, but lipid anchored maybe not so much, right? So there's 3 pictures over there on your left hand side of what they look like, but essentially they are just proteins that are hanging out with your phospholipids in your membrane. So integral, just like integration, right? Integration means you're like adding it, incorporating it in there. That's this guy right here, right? So you're actually sticking the membrane in or sorry, you're sticking the protein in the membrane, right? Peripheral, like your peripheral vision, is just kind of off to the side. The same thing. Peripheral means that it's either on top or bottom of your membrane. It's not inside, right? Lipid anchored, just like it says, there's a lipid that attaches it to the membrane. So self-explanatory. But let's take a look at it one by one. Here is a membrane protein, right? So integral, like I said, they're the ones that penetrate and pass through the membrane, right? The special thing about this one is that they have special hydrophobic domains, right? They have domains meaning like the pieces, right? Remember that from the last PowerPoint. They have hydrophobic domains that allow them to embed themselves in the membrane, right? So things like, it says it right here, things like channel proteins, right, are able to embed themselves in there and allow things like water to come in or ions to come in because they are providing like a tunnel, essentially. So here's a picture of what that looks like, right? There's different types of integral proteins, right? And you can see here in the middle, this kind of light purple color right here, right? Those are the hydrophobic regions. It's kind of like they're threading through the membrane, right? And then once you get to this part and this part, these are the ones that are hydrophilic because now they are sticking out, right? So hydrophilic, they're sticking out that and they are interacting with the liquids around them. So what these proteins do is that they twist their R groups in a way that the hydrophobic pieces stick out and interact with the hydrophobic tails, right? Now, I know I'm not answering the question right now. I just wanted to use this picture here really quick, right? But I'm gonna write it down. Hopefully you write it down too, right? The protein twists their R groups so that the hydrophobic portions are interacting with the hydrophobic tails, right? And there's several ways you can do that. Like this one that I just labeled on, this one only does it once. This one does it 3 times, right? This one does it 2 times, right? So they can embed themselves in there multiple times depending on what R groups they have available. Okay, let me see here. What common protein motif do you observe in this illustration? That's just what it is, right? That we have both hydrophilic and hydrophobic components in these integral membrane proteins, right? And what can you predict about the chemistry? You're talking about hydrophobicity here, right? Hydrophobicity. Okay, this is just another way to look at that, right? So you can determine what type of integral protein you are based on what's sticking out where. So yes, I'm going to put a star. Yes, you need to be able to figure that out. I could give you something that looks like this, and I might ask you, what type of integral membrane protein is that? And hopefully you'll tell me it is type 1. And just in case you're sitting there and you're like, what the heck is the N and the C? That is the groups that's attached to your protein, right? Remember, you have an amino group and a carboxyl group, right? So N is your amine group, and then your C is your carboxyl group. So depending on where it's sticking out, right? Here's the cytoplasm, which means this is inside the cell, and this is outside the cell. So depending on where that's sticking out and the length of it, right, you need to be able to tell me which one is which. So if you have any questions about that, let me know. All right, I'm going to skip to the next one. Okay, so that was integral. So our next one is peripheral. And just like I said, peripheral means it's like it's off to the side, right? So they are attached to the membrane by weak bonds and are easily solubilized and removed. So in this picture, the green one is the protein, right? This other one, this is something completely different that is literally like you're holding its hand, right? You're not actually in the membrane, you're just kind of holding onto it, and that's why it's easy for them to come off, because the bonds that are, the bonds that are like right here, right? You can break those easily, and that peripheral membrane can just go, right? So let's see. It says that it is associated with membrane surface by non-covalent bonds, right? By non-covalent bonds. And then the last type is your lipid-anchored membrane protein. And just like the name says, it is anchored by a lipid, which is this guy right here. Right? So it has this big word, and I'm going to take it one syllable at a time again. Glycophosphatidyl'inositol, GPI, link protein, right? And so what it does is it's using the lipid and then this structure right here, right, in order to hold on to the protein, right? The other type of lipid anchored protein is going to be this one, where it's literally, it has this long hydrophobic tail to it, right? And then it's literally just barely holding on is what it is, right? Literally barely holding on to the protein. So that's the only difference. Don't have to super complicate it, right? Just be aware that there's three main parts. The main idea here is that you need to be able to have something hydrophobic to attach yourself to the membrane, whether that's the protein itself or other things like lipids, right? Or other anchors that would be there too. Okay, so now that we know the main parts of a membrane, we're going to talk about fluidity for a little bit and really just hone in on the function of the membrane during the next lecture, right? So here's a phospholipid bilayer. And like I said earlier when we were talking about cholesterol, they do depend on a variety of different things, especially temperature. Right? So right here it says structure depends on temperature. So we have two types of phospholipids right now that we're looking at, right? One of them on the left is, I would say, fluid. There's space between them, and they seem to have a lot of movement around them, right? The one on the left, more rigid, right? They're all kind of straight. The legs are straight instead of curved, right? And they're all packed together. So if we're talking about saturation, this one would be unsaturated, this one would be saturated, right? But there's something called a transition temperature, which I'm going to talk about in the next slide. But once they hit this transition temperature, it changes the structure of your phospholipids. So the one on the left, that's going to be above the transition temperature, right? So above the transition above, I'm just going to call it TT, right? Above transition temperature. This one is below transition temperature. And the transition temperature is determined by a lot of different things, which is going to be on the next slide. Right? So here it is. It's the temperature at which the membrane is converted from a fluid state to a crystalline state. So if I go back here, and I'm just labeling here, right? This would be my, what did I call it? My fluid state. So here's fluid state. And this one is my crystalline state. So if I just come up with a random number, let's say 10 degrees, right? Celsius. So if I am above 10 degrees, above 10 degrees, that means it's warmer, right? So warmer means I become more fluid. And if I'm below the transition temperature, below 10 degrees, that means it's colder, which should make sense, right? And if you just think about things like butter, right? Butter is a saturated fat, right? If I put the butter in the fridge, it's gonna stay solid, right? Which means that it's saturated. If I take the butter out and leave it out for a couple hours in room temperature, it's going to start melting, right? So it's going to be more fluid. It's going to be like liquid butter. So that's the same analogy here. So this temperature, and it's again different for every single lipid bilayer, but this temperature depends on the type of lipids that it makes up. There's 3 of them if you remember. The saturation, right? Fatty acid length. Some of them are longer than others. in the presence of cholesterol. Right? So if I just break this down a little bit, because I think on your learning goals it wants you to list them, but I need you to be a little bit more detailed to it, right? So I need you to make a relationship with it, right? So you know, take a look at if it's saturated, what does that mean? If it's unsaturated, what does that mean? If it has high temperature, what does that mean? If it has low temperature, what does that mean, right? And if you need some extra guidance on it, your textbook gives you really great examples and really great explanation on there too, right? Okay, next is going to be cholesterol. So I said that we were going to expand on it a little bit more here. So cholesterol is like that backpack I told you. That's this example right here. See how it's right next to two of the phospholipids. It does help with the fluidity of the membrane. And then we have something called lipid rafts, which I took a look at what your textbook said about it, because sometimes it's different depending on the textbook. But these lipid rafts, they have things attached to it, and a lot of scientists are still kind of debating whether or not this shows up in living things, because they've only ever seen it extracted and they've never really actually seen it in a living organism. But what it is just literally it's a lipid, right? Like a bunch of phospholipids together that has something attached to it, like either a sphingolipid or some other stuff, right? And it kind of just floats around, right? And so what they think it's for is they think it's for signaling purposes. But like I said, they don't really know if it even exists in living cells. They've only seen it outside, right? Okay, last thing that I want to talk about with you guys in terms of the structure of a plasma membrane is their ability to fuse together. So things like vesicles, when you're transporting items around your cell, they are enclosed in a membrane, right? So this particular diagram, I think there's a similar one in your textbook. It's showing you a mouse cell, which they attached a fluorescent antibody to make it pink, and then the human cell, which they attached it. Did I say protein? They're attached to proteins. The antibodies are attached to proteins and they make it glow different colors. So the pink one is mouse and the blue one is human. And so they observe how they fuse together based on these fluorescent markers, right? And what they saw is that eventually they do come together, right? And then this is like the real like picture that you see. So you see that at a certain point, one part of it is where the human proteins are, and the other part is the mouse proteins. So what this tells us is that proteins and the phospholipids can actually move around. I don't know if you knew that, but your phospholipids and your proteins can move around. And this picture right here shows it a little bit, right? So they can rotate, just like this guy right here. They can move around, right? So for side to side, and then they can also flip-flop. Flip-flopping one is the rarest one though, right? They can do it, it's just it takes special enzymes to make them do that, and it just, I think, takes a lot of energy for it to do that, right? So they need a special enzyme to be able to flip to the other side of the membrane. So then I need you to then do some critical thinking, right? So based on what we talked about with membrane fluidity and based on what I told you about membrane fusion, I need you to think about what would happen to this experiment if it was conducted at 4 degrees Celsius, so just cold, right? If it was cold, how does that affect the membrane fluidity of the membrane? Right? How does that affect the membrane fluidity and the ability of proteins and phospholipids to move around? Right? And then the other thing is going to be predict the outcome if the cell membrane had a higher amount of cholesterol. So same thing. So on Canvas, there is going to be a quick little, I don't know what I'm going to call it. I'm going to make it right now, but there's going to be a little answer form where I need you to answer those questions just to the best of your abilities, right? And then we'll talk about it. We'll pick it up and talk about it on Wednesday. I just want to make sure and see if you got the main idea between the relationship of the two, right? You'll get credit either way. I just want you to think about it, right? And I will talk to you guys about that when I see you on Wednesday. Right, this is just another look at the phospholipid on the underside. So on the underside, there is cytoskeleton that hold on to some of these proteins. I know we talked a lot about anchoring it within the membrane, but on the underside of the membrane inside the cell, you do also have cytoskeleton that can hold on to them. So that's the end of this PowerPoint. Please don't forget to turn in that quick little assignment, and then I'll see you guys on Wednesday. Please bring your questions if you have any, right? I know it's very difficult for me to anticipate your questions when you guys are not right in front of me. So have a good one. I'll see you guys on Wednesday.